Buonasera, buonasera a tutti, buonasera a voi che siete qui in sala Buzzati e buonasera a tutti quelli che ci stanno seguendo sul sito di Corriere.it e sul sito della Fondazione Balzan. Sono Roberto Stringa, sono il direttore della Fondazione Corriere e insieme con Ferruccio De Bortoli, che oggi purtroppo non può essere con noi qui presente per un imprevisto dell'ultima ora, voglio porgervi appunto i saluti della nostra fondazione. La nostra fondazione è davvero felicissima di poter e onorata di poter ospitare l'annuncio dei vincitori dei premi Balzan anche per quest'anno, per il 16° anno consecutivo. Ormai questa è diventata quasi una sorta di ricorrenza, ma più che una ricorrenza io credo che questo rappresenti da un lato che per noi ogni anno l'attività, tutto il programma di attività che noi facciamo, tutta la nuova stagione, parte sempre da questo punto fisso che abbiamo appunto ogni stagione. E la seconda cosa forse vuol anche dire che questa nostra casa, questa casa della parola, Sala Buzzati, in qualche modo, oltre a essere la nostra casa, è diventata, o forse è sempre stata, la casa di Eugenio Balzan e della sua fondazione. Quindi con l'augurio anche per il prossimo anno e per tutti gli anni futuri di rivederci e ritrovarci sempre qui per l'annuncio dei premi, ora do la parola ad Anna Scavuzzo, vice sindaco di Milano.
Buonasera a tutti, buonasera a tutte. Intanto grazie perché si rinnova questo patrocinio che il Comune di Milano accorda sempre volentieri al percorso certamente di premio, ma di attività di Fondazione Balzan, a questa collaborazione con Fondazione Corriere, che è una collaborazione che ci onora e ci lusinga perché riporta un po' alle radici del nostro essere milanesi, con questa capacità di essere ben legati alla nostra comunità, ma con uno sguardo aperto e ottimista al mondo, e questa capacità di errorare con quelle che sono anche le nostre più belle, importanti eccellenze, quella che è una visione ottimista del futuro. E credo che questo credere nella scienza, consolidare il rapporto con la comunità scientifica, affidare alla scienza un messaggio di pace che travalica i confini e che fa sì che le eccellenze siano al servizio di tutti e di ciascuno in tutto il mondo, penso che sia il messaggio più bello che soprattutto oggi la comunità umana ha bisogno di rinnovare, e quindi che sia un messaggio di fiducia, sia un messaggio che investe sul futuro a partire dalle comunità scientifiche di tutto il mondo e dalle loro eccellenze e sui loro giovani e le loro giovani. Credo che davvero siano dei messaggi che anche alla nostra comunità milanese dicono tanto rispetto a quello che vogliamo essere anche noi nel futuro. E da ultimo un pensiero particolare, perché sul palco ci sono 2 donne e che nella loro esperienza e nella loro vita sono state anche per noi, comunità politica, comunità cittadina, comunità italiana, esempio di grande capacità di mettersi al servizio della comunità. E credo che anche questo sia un segnale bello di continuità rispetto a un'intuizione che quella di Fondazione Balzan da tempo porta avanti, di far sì che davvero la comunità cresca con l'esemplarietà delle persone che si mettono al servizio di tutti. Buon lavoro e auguri e congratulazioni a tutti coloro che verranno premiati. Grazie.
Molto carino, molto carino. Bene, buonasera a tutti. Io sono la nuova neopresidente della Fondazione Balzan Premio e oltre a salutare tutti volevo ringraziare voi della vostra presenza, ringraziare la Fondazione del Corriere della Sera con il dottor Roberto Stringa che si fa l'attore anche verso Ferruccio De Bortoli, la Presidente, perché abbiamo una terza Presidente che organizza questi premi, che è Giselle Girgis, che è qua, la Presidente della Fondazione Premio. Tutto il Comitato Generale Premi, ne conoscerete alcuni che oggi, fino a un po' di tempo fa, e ringrazio il professor Mariani per quanto ha fatto, ma oggi viene magistralmente condotta dalla professoressa Cartabia, che ringrazio. Questa fondazione lavora anche a stretto contatto con altre due accademie, l'Accademia dei Lincei, il professor Roberto Antonelli, da cui andremo a novembre, e l'Accademia Svizzera delle Scienze, il professor Marcel Tanner, che anche ringrazio moltissimo. E mi sono tenuta per ultimo il nostro consiglio con la grandissima vicepresidente Laera, con il professor Giuseppe Vegas che ci ha appena raggiunto come delegato del governo, e il ringraziamento particolare, come potete immaginare, al consiglio va al professor Quadrio Curzio, che indegnamente sostituisco. Che dire del professor Curzio? Semplicemente che da tempo mi ha fatto da guida perché innanzitutto per la sua conoscenza e competenza, ma soprattutto perché ha un'indole decisamente vivace e che guarda molto al futuro, che non si ferma mai, che guarda sempre all'innovazione e che ha questa lungimiranza. E quindi penso che già prima mi è stato di guida e penso che sarà di guida anche nella conduzione di questo Consiglio. Una lungimiranza che era anche quella della Fondazione Balzan, che è stata fondata quasi 70 anni fa, nel 1956, e ha iniziato a dare premi, importanti premi, più di 60 anni fa, nel 1962. E perché è stata lungimirante? È stata lungimirante nel supportare la ricerca, la scienza e le scienze morali attraverso il premio alle persone che sono protagoniste di questa ricerca. I premi infatti si sono susseguiti nel tempo senza modificare mai il metodo rigoroso della scelta delle materie e dei candidati vincitori, ma adeguandosi continuamente alle nuove classificazioni, perché non sta ferma certo la scienza. Quindi classificazioni dei saperi nuovi che conducono sempre di più verso un'interdisciplinarietà di cui abbiamo parlato anche oggi quando sono stati assegnati i premi. In generale quello che vogliamo fare è quello di evidenziare il ruolo trainante e catalizzatore della scienza. E questo significa riconoscere un metodo che è quello scientifico, sperimentale, che è trasparente ed è ripetibile, la principale modalità per realizzare responsabilmente e in maniera equa e solidale un futuro migliore e inclusivo. Ma significa anche ampliare il significato del fare ricerca, oltre gli specialismi settoriali. Questo è molto importante per noi. Il premio che noi diamo può stimolare, per esempio, un dialogo più continuo e maggiore fra le scienze umanistiche e le scienze cosiddette dure, fra la componente umanistico-letteraria e quella scientifica, senza tralasciare le scienze sociali ed economiche, che sono tanto importanti per il nostro paese. Perché un non dialogo fra queste culture diverse condurrebbe a un reciproco impoverimento e oscurerebbe quello che la scienza può fare. Infatti anche nelle varie materie che oggi si studiano, nelle varie discipline, si creano delle nuove discipline. Pensate alle scienze dei materiali che questa fondazione premia. Le scienze dei materiali cosa sono? La chimica, la fisica, sono tante materie che messe insieme riescono a dare un significato diverso. Anche la bioinformatica, in fin dei conti, sta seguendo questo percorso. In ogni caso, qualsiasi classificazione, qualsiasi categoria noi diamo alla scienza in senso lato, il premio di oggi è il riconoscimento del lavoro di scienziati, a qualsiasi categoria questi appartengono. Il valore del premio riflette l'importanza che viene data alla scienza e alla cultura attraverso due aspetti principalmente. Il primo è tangibile ed è l'aspetto economico con i 750.000 franchi svizzeri, che riflette in maniera rilevante l'importanza che diamo a questo premio e la qualità che vogliamo che questo premio tenga. Il secondo è intangibile, non per questo meno importante, perché riconosce a scienza e cultura un ruolo fondamentale nella società attraverso i suoi attori principali, ovvero le persone senza distinzioni di nazionalità, di razza e di religione. In un periodo come l'attuale, che è particolarmente devastato da conflitti, dove si tende sempre più a una polarizzazione dei principi che regolano la società, la vera sfida per la conoscenza è quella di fornirci strumenti e motivi per restare umani. E questo ci ricorda un manifesto del 1955 di Bertrand Russell e Albert Einstein, dove loro dicevano restare umani, o meglio sani di mente in un mondo di folli. Loro la auspicavano in un momento particolarmente difficile in cui c'era il pericolo, un pericolo di guerra nucleare, un pericolo costante, e invitavano, questa è la cosa ancora attuale, a considerarsi non indipendenti da etnia, credo politici, religiosi, ma a considerarsi solo come membri di una specie biologica che ha avuto una ragguardevole storia e di cui nessuno desidera la scomparsa. Ora questo manifesto torna un pochino nei giorni nostri, ma torna anche un altro aspetto dell'importanza del restare umani, che è la nuova sfida che dobbiamo affrontare, che è caratterizzata dalla rivoluzione digitale. La rivoluzione digitale che è quella che sta mettendo l'uomo e le sue capacità sullo stesso piano della macchina e dei suoi servizi. In questo senso il ruolo della società e dello scienziato non lo esime dal problema etico della responsabilità dell'uso delle sue scoperte, ma lo rende protagonista. E quindi oggi premiare gli scienziati vuol dire anche riconoscere questo ruolo, dando loro fiducia, credibilità, perché è un premio che si possono portare in tutto il mondo, perché possano continuare a fare ricerca, a trasmettere ai giovani, perché è molto importante il ruolo dei maestri, è fondamentale, e a usare il loro talento anche per favorire un mondo che vada verso la pace. Grazie. Passo la parola alla Presidente del Comitato.
Grazie, colgo anch'io l'occasione per ringraziare la Presidente Messa, ma soprattutto per la fiducia che mi avete accordato nel coordinare il lavoro di questo gruppo di persone, di studiosi veramente eccellenti che lavorano alla individuazione delle materie e dei premiati ogni anno, portandoci dei nuovi nomi e delle possibilità anche di ascoltare lectures e interventi di livello altissimo. E qui ho l'onore di svolgere il ruolo di annunciare i premiati ufficialmente, il primo dei quali è nell'ambito della giustizia riparativa, a proposito di discipline, di saperi che si stanno sviluppando. La giustizia riparativa è forse il più nuovo di tutti gli sviluppi dell'ambito giuridico, conosciuto anche nel nostro ordinamento proprio per un intervento del 2022. E il premio Balzan per la giustizia riparativa quest'anno è stato attribuito a John Braithwaite, australiano, professore emerito dell'Australian National University, con la seguente motivazione: per il suo contributo allo sviluppo teorico e alla diffusione della prassi della giustizia riparativa contemporanea, per il suo impegno a servizio delle istituzioni e della costruzione sociale, per il suo lavoro di alta divulgazione scientifica ed editoriale, per la sua dedizione alla crescita di culture più giovani, nelle più giovani generazioni, nei valori della giustizia riparativa. E ora, cambiando cappello, mi trasferisco al podio perché ho l'onore di poter leggere a tutti voi la laudatio, le motivazioni più approfondite per le quali il Comitato ha scelto il professor Braithwaite, uno dei fondatori appunto della giustizia riparativa, come premiato di quest'anno. Champion of Restorative Justice è il titolo di uno degli innumerevoli riconoscimenti civili e accademici attribuiti a John Braithwaite, e la qualifica di champion della giustizia riparativa davvero non è esagerata in questo caso. Ma che cosa è la giustizia riparativa secondo Braithwaite? Ricordiamolo con le sue parole. Restorative justice restores victims, restores perpetrators, restores communities. It is about the idea that because crime hurts, justice should heal. La giustizia riparativa nasce dalla constatazione che il reato è esercizio di potere e di sopraffazione sulla vittima, è un'azione violenta che provoca una ferita, molto spesso fisica ma anche materiale o morale, e la giustizia deve innescare un processo di guarigione. La giustizia riparativa si affianca perciò alla giustizia tradizionale che si amministra nei tribunali e si svolge attraverso l'incontro, è detta anche da molti la giustizia dell'incontro. Abbiamo fra noi Claudia Mazzucato che ha scritto insieme ad altri amici, Adolfo Ceretti e altri, un libro proprio su questo. Un incontro che è liberamente accettato al momento opportuno tra le vittime, gli offensori ed eventualmente altre persone significative per gli uni e per gli altri. Con l'aiuto di un mediatore si affrontano domande molto semplici ma decisive per le persone coinvolte. Cos'è accaduto? Perché quel fatto? Quali persone sono state colpite? Perché proprio loro? Cosa si può fare per dare prospettive future a tutti i soggetti coinvolti? La giustizia riparativa non va confusa con un atto di clemenza. Il lavoro di John Brathwaite l'ha evidenziato molto chiaramente, perché essa richiede all'offensore di assumersi tutte le proprie responsabilità davanti alle vittime e alla comunità coinvolta. Reintegrative shaming è il concetto elaborato da John Brathwaite in proposito. 30 anni di pratica della giustizia riparativa contemporanea e di studi su di essa mostrano che questi incontri danno un grande sollievo alle vittime, poi l'idea di ristoro, restorative appunto, ed è importante sottolineare, portano a una significativa diminuzione della recidiva attraverso la reintegrazione sociale degli offensori. John Brathwaite è uno dei fondatori degli studi e della prassi della giustizia riparativa in epoca contemporanea. Senza la sua opera, la giustizia riparativa non avrebbe guadagnato né la necessaria credibilità accademica né la fiducia delle istituzioni internazionali che ora la promuovono e la supportano ad ogni livello. Alcuni dei suoi concetti sono divenuti di dominio generale. Sottolineo in particolare l'idea di responsive regulation, che allude a una forma di regolamentazione responsiva che sa valorizzare i comportamenti positivi dei consociati che fin dall'inizio o a valle di un illecito decidono di osservare volontariamente o tornano ad osservare volontariamente le regole. L'ampiezza delle pubblicazioni di John Brathwaite è sconfinata, sia per interessi coltivati che per il numero di volumi e di articoli pubblicati, ma soprattutto ampia è la comunità di studiosi, di giovani generazioni che in tutto il mondo diffondono la cultura e gli studi sulla giustizia riparativa. Ma c'è di più. John Brathwaite non si è mai chiuso nella sua torre d'avorio. Ha speso moltissime energie nella costruzione di realtà sociali e di una società civile dove la giustizia riparativa potesse essere praticata. Ha contribuito in modo decisivo a sorreggere questa prassi sociale con un'architettura normativa e istituzionale riconosciuta che potesse dare vita a un sistema complementare alla giustizia tradizionale. Negli anni più recenti il professor Brathwaite ha sviluppato la riflessione su questo ambito in relazione ai grandi problemi della nostra epoca: sostenibilità, guerra, pace, cambiamento climatico, finanza, salute. E a questo proposito, proprio con il pensiero rivolto ai grandi conflitti del nostro tempo, merita una particolare sottolineatura il suo progetto, iniziato ormai da molti anni e proiettato ancora per molti anni a venire, di Peace Building Compared. Studia i grandi conflitti del nostro tempo e i processi di ripristino della pacificazione per mettere in evidenza gli elementi di successo anche a fronte dei grandi conflitti armati che affliggono il nostro tempo. Insomma, per utilizzare le parole, le idee stesse del professore che noi stiamo premiando, la giustizia riparativa è una strada fruttuosa e semplice, come dice il titolo del suo ultimo libro, percorribile sia per i grandi dilemmi del nostro tempo, sia per le piccole ma brucianti conflittualità che affliggono la vita quotidiana di ognuno. E ora ho il piacere e l'onore di chiamare sul palco Peter Kuhn, che enuncerà le ragioni del premio Balzan 2024 per la storia della scienza moderna e contemporanea, che è stato attribuito a Lorraine Duston, direttrice emerita del Max Planck Institute della Scienza, per la storia della scienza, con la seguente motivazione: per l'ampiezza, l'originalità e la varietà del suo lavoro, che ha saputo evidenziare le rappresentazioni mentali e i valori alla base dell'attività di ricerca in una vasta gamma di campi del sapere, per il numero e la qualità dei suoi articoli e dei suoi libri che hanno aperto nuove strade nella storia dell'epistemologia delle scienze, per il suo contributo alla formazione di generazioni di ricercatrici e ricercatori e per il suo sostegno in qualità di responsabile di prestigiose istituzioni a progetti di ricerca innovativi. Mi piace sottolineare che in entrambi i casi abbiamo voluto valorizzare anche proprio il ruolo di mentori di questi grandi professori, ricordando che parte della grande somma che il premio Balzan riconosce è anche devoluta per progetti futuri da coltivarsi attraverso il contributo di giovani generazioni. Grazie Peter.
Buonasera. Il filo rosso che collega gran parte dell'opera di Lorraine Duston, direttrice emerita dell'Istituto Max Planck per la storia della scienza a Berlino, è la costante sottolineatura dell'analogia tra i concetti e paradigmi che hanno scandito l'evoluzione del pensiero scientifico dell'età moderna e contemporanea e quelli sui quali si è fondato lo sviluppo storico di altri ambiti di conoscenza e delle forme di organizzazione della società. È un tratto caratteristico che traspare fin dalla sua prima monografia, Classical Probability in the Enlightenment, in cui ricostruisce la nascita nel Settecento di una scienza predittiva basata su metodi matematici rigorosi. Questa scienza, cioè il calcolo delle probabilità in un senso odierno, nasce in un ambito culturale che richiedeva di calcolare i rischi di perdita e di guadagno sia nel gioco di azzardo sia nell'assicurazione di una nave o di una vita. La longue durée dell'impegno dell'autrice in una concezione della storia della scienza come parte integrante della storia globale della conoscenza trova conferma nel più recente dei suoi volumi, Rules, del 2022, nel quale illustra l'emergere e la continua crescita del concetto di legge, di regola, non solo nelle scienze della natura, ma in ogni ambito della cultura e delle pratiche sociali. Lorraine Duston mette in forse la tesi dello splendido isolamento delle discipline scientifiche per sottolineare invece le connessioni a livello epistemologico con le scienze sociali e umanistiche. Questo approccio anima una serie di saggi penetranti su categorie e pratiche fondamentali della ricerca scientifica, come per esempio la natura, la razionalità, l'oggettività, lo sperimento, il dato di fatto, la prova e così via, dimostrando che queste nozioni sono sorrette da rappresentazioni mentali e valori condivisi che cambiano nel corso della storia. Per dare un esempio, in Objectivity, un libro del 2007, scritto in collaborazione con Peter Gallison, la Dustin, attraverso l'analisi di una serie di casi storici, fa toccare con mano come le perentorie affermazioni del carattere oggettivo delle teorie professate dagli uomini di scienza, soprattutto uomini, si fondino spesso su generalizzazioni non corroborate da riscontri osservativi sperimentali, su opinioni preconcette sulla costituzione della natura e sui suoi fini, su credenze religiose e sui condizionamenti dell'appartenenza a comunità di riferimento in competizione con altre. Altri saggi illuminanti sono dedicati al processo che segna il passaggio dal predominio di ricerche condotte individualmente e caratterizzate da forte spirito competitivo e da estrema riservatezza, all'imporsi sempre più marcato dalla metà del XVIII secolo di reti internazionali di collaborazione per la realizzazione di progetti che richiedevano cospicui sostegni finanziari e infrastrutture sofisticate. Esemplari da questo punto di vista i saggi raccolti nell'ultimo suo volume Rivals, How Scientists Learned to Cooperate. Partendo dalla rievocazione delle prime espressioni concrete della consapevolezza dei benefici dell'unione delle forze, cioè le spedizioni organizzate a fine del Settecento per osservare da diversi luoghi del pianeta il transito di Venere, l'autrice analizza le circostanze che segnarono l'avvio e le dinamiche di sviluppo di due giganteschi progetti di ricerca fondati sulla cooperazione e sulla contribuzione finanziaria internazionale, la Carte du Ciel, concepita alla fine dell'Ottocento, e l'International Cloud Atlas, avviato dopo la Seconda Guerra Mondiale. Last but not least, Lorraine Dustin è una brillante scrittrice che riesce a toccare un pubblico che va al di là dello stretto circolo degli specialisti, senza però sacrificare né precisione né profondità scientifica. I suoi saggi e libri sono godibilissimi. Mi sia concesso un ultimo rimando al volume Wonders and the Order of Nature, scritto assieme a Catherine Park, che trasporta il lettore in un viaggio affascinante che, partendo da un medioevo popolato da mostri, prodigi, miracoli, lo conduce al completo ribaltamento di questa visione del mondo con l'affermazione illuministica di una natura governata da leggi che non ammettono eccezione. Così vorrei terminare la mia laudatio con un invito alla lettura della vincitrice del premio Balzan 2024 per storia della scienza moderna e contemporanea.
Grazie caro Peter, passiamo ora ai vincitori delle scienze dure. Il primo vincitore che ho il piacere di annunziare per la materia, per l'ambito materiali nanoporosi per applicazioni ambientali. Abbiamo cambiato l'ordine, non c'è nessun problema. Meccanismi biologici dell'invecchiamento è Michael Hall, Svizzera, USA, professore di biochimica a Basilea, con la seguente motivazione. Per i contributi innovativi alla comprensione dei meccanismi molecolari che regolano la crescita cellulare. Michael Hall ha scoperto due proteine, TOR1 e TOR2, che regolano la crescita cellulare e il metabolismo in risposta ai nutrienti. Queste svolgono un ruolo centrale nel processo di invecchiamento e nello sviluppo di malattie legate all'invecchiamento, come il cancro, il diabete e le patologie cardiovascolari. La parola a Sebastian Bonhoeffer per la laudatio. Grazie.
Grazie, buonasera signore e signori, e con questo il mio italiano già finisce. So, good evening, ladies and gentlemen. It's a great honor and a privilege to say a few words towards the foundational impact that this year's laureate has had on elucidating the mechanistic basis of aging. Well, all organisms age, from the smallest to the largest, from bacteria to blue whales. And so it is a fundamental question, why do we age? And what is the mechanistic basis of this aging? And these are really deep fundamental questions that I think attract the human curiosity, the curious minds, but they're also very important questions in the sense that they really have far-ranging medical implications. So let me recount a little bit the exciting story how Michael Hall contributed to our understanding of the biological mechanisms of aging. It all starts with a drug called rapamycin. Rapamycin is an immunosuppressive drug, which also has cytostatic, i.e. anti-cancer activity. Rapamycin is a small molecule that was isolated from a soil bacterium of the genus of Streptomyces, and the bacterium was discovered in an expedition to an island, Rapa Nui, which is better known to many of us as Easter Island, and that's how the drug got its name, rapamycin. And now the story with Michael Hall's contribution and groundbreaking discovery starts in the early 1990s when he decided to search for the molecular target of this drug rapamycin. And in doing so, he discovered two genes which are referred to as TOR1 and TOR2. TOR just stands for target of rapamycin. He then continued to aim and elucidate the cellular function of these TOR genes, and in a series of truly groundbreaking and seminal papers, he demonstrated that the function of these TOR genes and proteins is to regulate cell growth by sensing nutrient availability. This caused a real paradigm shift in the field because up to that point, the consensus view was that cell size was limited by nutrient availability and not really itself actively regulated. The fact that then it was discovered by Michael that TOR is a master regulator of cell growth and metabolism in response to nutrients really fundamentally transformed our understanding of cell growth and then in consequence led to molecular understanding of many of the growth-related cellular processes, including aging. Michael Hall discovered that TOR1 and TOR2 form two large protease complexes. That's just one of those. They are now known as mTORC1 and mTORC2, where the M stands for mechanistic, and these are structurally and functionally distinct. Importantly, these complexes are evolutionary conserved, which means we find them from yeast all the way to humans, and that also means that the findings have broad implications. These complexes match cell growth to the nutrient supply and many external signals, including hormones, by controlling the balance of activity of both catabolic, i.e. degradative processes, as well as anabolic constructive metabolic pathways. mTORC regulates cell growth and metabolism by controlling the synthesis of cellular components like proteins, fats, nucleic acids that are important for cell growth. But importantly, mTORC also inhibits autophagy, as is indicated there. Autophagy is the process by which waste material in cells gets degraded, and impaired autophagy can be detrimental to cells and lead to age-related diseases like diabetes and others. In contrast, this other complex, mTORC1, regulates cell proliferation and survival. So Michael Hall's work has had multiple impacts on the study of aging. Concretely, his discovery that TOR1 is inhibited by rapamycin explains why rapamycin extends lifespan in multiple animal models. In fact, one of the key findings in the field that existed for a long time is that caloric restriction has an effect in terms of extending lifespan. And his groundbreaking demonstration that nutrients activate TOR1 now provides a mechanistic explanation for how these dietary restrictions actually work in extending lifespan. This works essentially because we have this inhibiting effect of mTORC1. Dysregulation of this process also results in many diseases of aging, including cancer, diabetes, and cardiovascular disease. Now, if I summarize, Michael Hall's groundbreaking discovery of tau proteins founded an entire new field that has now become extremely large. He has been at the forefront of this for over 30 years. Michael Hall's studies have provided a mechanism for a lifetime extension by dietary restriction, and his work has helped to elucidate fundamental and clinically important mechanisms in aging-related biology. And I think his work carries the hope that these fundamental insights may eventually also help to extend healthy human lifespan. Thank you.
Now we are, no, sorry. Ora possiamo ritornare alla materia che avevamo annunciato erroneamente precedentemente, materiali nanoporosi per applicazioni ambientali. Il vincitore è Omar Yaghi, attualmente all'Università di California, Berkeley, per la seguente motivazione: per i rivoluzionari contributi alla scoperta e allo sviluppo di materiali a struttura nanoporosa e per l'avanzamento delle loro applicazioni nella cattura del carbonio, nell'immagazzinamento dell'idrogeno e nell'estrazione dell'acqua dall'aria del deserto. Yaghi ha sviluppato principi fondanti di progettazione fondamentali e innovativi metodi di sintesi, creando due ampie classi di materiali nanoporosi: le strutture metallo-organiche MOF e le strutture organiche covalenti COF. Questi materiali pionieristici sono ora in prima linea negli sforzi globali per affrontare le sfide critiche di sostenibilità e ambiente che il nostro pianeta sta affrontando. Maria Strömme ci dirà qualcosa di più sulle ragioni della premiazione. Prego Maria.
So thank you. Everything sounds so much more beautiful in Italian. And you probably said the first part of what I'm going to say, but now you will hear it in English. So, ladies and gentlemen, it's a great honor for me to speak about this year's Balzan Prize laureate in the field of nanoporous materials for environmental applications, Professor Omar Yaghi, as you can see here. Professor Yaghi's groundbreaking work in the field of nanoporous materials has not only transformed the landscape of chemistry, but has also provided critical tools to address some of the most pressing challenges facing our planet today. His pioneering contributions have led to the discovery and development of, like you said, metal organic frameworks, which we say call MOFs, and covalent organic frameworks, which we call COFs. These materials are now at the forefront of global efforts in sustainability and environmental preservation. So MOFs, you can see one MOF structure here. They are made of metal ions connected by organic molecules, forming a highly porous structure. So they can store gases, they can filter chemicals and serve in catalysis due to their large surface area and tunable properties. COFs are similar, but they are made entirely of organic molecules connected by strong covalent bonds. They also have a porous structure and are used in applications like gas storage, filtration, and energy storage. Only 1 gram of these materials can have an internal surface area of 6,000 to 7,000 square meters, which is the same area as an entire football field. And this is important to understand their enormous capacity to absorb materials we want to get rid of from our environment and to store valuable materials we want to keep. Omar Yaghi is widely recognized as the founder of reticular chemistry, a field that has revolutionized our understanding of how chemical structures can be assembled from molecular building blocks. This approach, which many once thought to be chemically impossible, has led to the creation of materials with extraordinary properties. And these materials, known for their ultra-high porosity, they have opened new horizons in clean energy storage and generation, including the storage and separation of hydrogen, methane, and carbon dioxide. Moreover, they have advanced applications in clean water production, supercapacitors, and conductive systems. The journey of Professor Yagi's remarkable discovery began in the early 1990s when he proposed the radical idea of connecting metal ions with organic linkers to form strong, ordered crystalline structures. Despite skepticism from the scientific community, Professor Yagi persistence and vision bore fruit in 1995 when he successfully created the first metal organic framework. This achievement, published in Nature, paved the way for a new class of materials with unparalleled surface area and stability, characteristics that are crucial for a number of clean tech applications. One of the most notable milestones in his career was the development of MOF5, that you can see the structure of here, a material with record-breaking porosity and robustness, which became a cornerstone for the fields of MOFs. I should tell you that now more than thousands of MOFs have been made by several researchers, but this is the most famous one. This discovery of MOF5, also published in Nature, highlighted the immense potential of MOFs, showing the world that these materials could indeed harness the power of gases and molecules for clean air, clean energy, and clean water. Not resting on his laurels, Professor Yagi continued to push the boundaries of chemistry by developing the COFs, the covalent organic frameworks, in 2005. Here you can see the most famous one. They're composed entirely of organic molecules. COFs are held together by the strongest bonds in chemistry, covalent bonds, making them highly durable and uniquely structured for a number of applications. So, the materials created by Professor Yagi's innovative research have set new standards for what is possible in the realm of chemistry. They are now critical tools in our global fight against climate change, offering efficient and sustainable solutions to capture carbon, store energy, and harvest water, even from the driest environments. And the Balzan Prize recognizes not just scientific achievements, but also the potential of the work to impact humanity in profound and lasting ways. Professor Omar Yagi's contribution, they exemplify this ideal, demonstrating how pioneering science can lead to practical, life-saving solutions for our world's most critical challenges. So in conclusion, Professor Yagi's work is a testament to the power of innovative thinking, perseverance, and the relentless pursuit of knowledge. So it is fitting today that we celebrate his achievements, which have paved the way for a more sustainable and prosperous future for all, hopefully. Thank you.
Thank you, Maria. Ora è il momento di annunciare le materie che saranno premiate nel 2025 secondo le consuete modalità. Storia dell'arte contemporanea, Scienze dell'antichità, democrazia ateniese rivisitata, Atomi e misura ultraprecisa del tempo, terapia genica o con cellule geneticamente modificate. Come ogni anno ciascun premio ammonterà a un valore di 750.000 franchi svizzeri ciascuno e come ogni anno si prevede che metà della cifra, come ho anticipato poco fa, sia poi devoluta allo sviluppo di programmi anche attraverso la collaborazione di più giovani generazioni. E con questo ho concluso il mio compito,
Non
ti
è
restituisco la parola.
Non è detto
Come?
che tu abbia concluso. Non è detto perché adesso è il tempo per le domande, quindi se ci sono domande dal pubblico io spero che il messaggio delle motivazioni di questi premi vi abbia, come dire, convinto o comunque che sia ben comprensibile per tutti, ma magari ci sono domande anche su queste materie che sono state scelte per il 2025. Io non vedo bene, quindi dovete...
Ti aiuto a
alzare. Ecco,
okay.
Grazie.
Grazie.
Molto gentile.
Direi di no.
Dai. Una domanda. Prego. Là.
Sì.
No, vorrei sapere, ogni anno cambiano le materie, cioè vengono premiati a ambiti diversi.
Sì.
Ma in base a cosa? Qual è il criterio in base si decide?
Direi che sono un po' anche le urgenze della nostra epoca che attirano l'attenzione su ambiti o settori di studio. Molto spesso non è una materia nella sua interezza che viene premiata, ma viene indicato un particolare ambito di studi. Faccio un esempio sui i settori che conosco meglio, che sono quelli umanistici e delle scienze sociali. Quest'anno sarà premiato nell'ambito degli studi classici il tema della democrazia. Ora è evidente che il tema della democrazia è un tema che sta occupando e preoccupando sotto profili diversi l'opinione pubblica, ma anche molti studiosi. Nell'ambito delle scienze costituzionalistiche che meglio conosco, scienze politiche, sono innumerevoli gli studi ormai sul regresso delle democrazie, sulle crisi delle democrazie liberali e così via. La scelta è evidentemente dettata da un interesse molto particolare che si impone in modo significativo, così come negli scorsi anni, l'invecchiamento, le problematiche ambientali, e invita ad andare a guardare secondo i canoni però degli studi accademici, quindi che vanno in profondità, non inseguono soltanto i problemi dell'attualità, però partendo da problemi attuali andare a individuare degli ambiti che ci possano offrire degli strumenti e degli approfondimenti per guardare il nostro tempo con una prospettiva di lungo periodo che può partire dal passato, ma soprattutto che si proietta nel futuro.
Grazie mille, dottoressa.
Grazie.
Ci sono altre curiosità, domande?
Teniamo le energie per la lezione.
Sì, direi di sì. E quindi ringraziamo di questa presentazione, facciamo i migliori auguri ai vincitori e è il momento della lezione. La lezione che viene tenuta da un vincitore del 2023 che sarà estremamente interessante, Jean-Jacques Hublin. Thank you for coming, grazie di essere qua oggi, e lui ci parlerà appunto dell'argomento in cui ha vinto principalmente nella scorsa edizione della Homo sapiens replace Neanderthals in Europe. Luckily, hopefully. Grazie.
Grazie. I'm going to speak in English like some of the other speakers. Unfortunately, my Italian is very limited. I'm very honored and very pleased to be invited to give this lecture for many reasons. First of all, I'm very grateful to the Balzan Foundation for awarding me the prize last year and supporting my research. And I'm also very glad to be invited at the Fondazione Corriere della Sera, and especially in this room that is named Buzzati, because Dino Buzzati has been an author that counted a lot for me when I was a young man in my personal development. So today I want to speak about this main event in human evolution, which is the replacement by populations of Homo sapiens of the Neanderthals in Europe. And you may wonder why we care about that. You know, after all, yes, Neanderthals disappeared and we are here. And I think it's actually a very important question to address because what we try to understand is not so much things about Neanderthals, but things about ourselves. And I think in this story, I hope you'll see that finally what is unveiled somehow is features of our own species that are very unique and, as you know, in today's world have a lot of effects on the environmental conditions and the future of our planet and ourselves. So humans today are one species, and I think you don't have many problems to identify what humans are. They're all over the place. We are more than 8 billion now, and humans are a very homogeneous species. There are differences from one part to the other of the world, but these differences are really, I would say, cosmetic. It's about skin color and some other features, some other variation. But altogether, humans represent a very homogeneous species. And this is related to the fact that all the humans living today have a common ancestry, which is not very far in the past. It's a rather recent ancestry. So we diverge, but not much. And this situation of having one species of humans that has colonized all the planet may look to you like a normal situation, you know. It's not. And if you move in the past, not a long time ago, just 40,000 years ago, you may think 40,000 years ago, it's a long time. It's not such a long time compared to the 7 or 8 million years of evolution of hominins, the ancestors and the relatives of humans. So if you move in past just to look at what has been going on during the last million years, you see that there are many different species of humans, and it's something that to us is a bit difficult to sort of grab, you know, the notion that there are humans of a different species. Like today there are chimpanzees and gorilla and orangutans, different species of large apes. In the past there are different species of humans. And one of the most known is the Neanderthals, but as you see there are many others. And if you look at this diagram, you see that the only one that reached the top, which is representing the present, is this orange bar, which is the species of Homo sapiens that developed in Africa, when all the others developed in other parts of Eurasia, and especially the Neanderthals. And these species of humans were very different one from the other sometime. We're not talking about this kind of differences that you can find today between humans living in different continents. We're really talking about huge differences. One example that's extreme is represented on the right side, and you see a reconstruction of a little creature called Homo floresiensis that had a brain about like a grapefruit, and that lived in an Indonesian island until like 50,000 years ago. And this is really something very different from what you would call a human. And the story is that with the development of these different groups of humans, eventually, and this is a rather complex phenomenon, but starting about 180,000 years ago, we start having humans, Homo sapiens, that had evolved in Africa, spreading all over Eurasia, eventually reaching Australia and other lands where there were never hominins before, like the coldest lands of Eurasia, and eventually colonizing America. And this is probably the most important event of the last million years in terms of human evolution. It maybe is the most important event at all, as a whole, in hominin evolution. Because it never really happened before. And it's something we would really like to understand because, of course, there is a whole literature, many scientific documentaries about why the Neanderthals got extincted, why other groups got extincted. In fact, the real question is why have we been so deleterious to other species. And we led to extinction all these groups, and not just groups of hominids, but also many other species of vertebrates, and especially mammals. What we know is that this expansion and this replacement is not a complete replacement. We know that there have been some hybridization between these Homo sapiens moving out Africa and local hominids, and especially in Europe, this has been well studied with the Neanderthals. What you have here is a representation, very crude, of your chromosomes. And so the 23 pairs of chromosomes, and you see little red bars on these chromosomes, and these little red bars represent Neanderthal DNA that is observed in present-day humans outside of Africa. And in this room, all of us, almost all of us, have about 2% of Neanderthal DNA. But you see that there are places in the genome where you never find a red bar. They are completely blue. So first of all, what you have represented here is all the places in the DNA where you could find Neanderthal DNA, but no one has all this DNA present, of course. This is putting together all the 2% of everybody. And when you do that, in fact, you don't obtain 100% of the Neanderthal DNA. You obtain this, which is about 40% of the Neanderthal DNA. And it means that there are places in the genome where it was bad to have Neanderthal DNA. And so the individuals who had the bad number in this genetic lottery, they never had descendants, and they did not reproduce. Maybe they even did not get born at all. So this story of the big replacement, we would like to elucidate it, but in fact, in most of the places in Eurasia, we don't have a lot of data, a lot of information about how it happened. And of course, we would like to know the details of the scenario and why it happened and how it happened. And in fact, the only place where more or less we can try to reconstruct this story is Europe. And it's Europe. Why? Because Europe is the place where paleoanthropology was born in the 19th century. And so for more than a century and a half, we have collected a lot of data. And this is one of the first Neanderthals that has been ever found in Europe. It was found in Gibraltar in 1848. And since then, a lot of fossils and a lot of archaeological sites have been studied. And so we have, I would say, in Europe, a better, a more detailed picture of what we have elsewhere. And this is why we can try to figure out what happened. So the Neanderthals, they are known in Europe, as I said. They have been also discovered in the Levant, in the Near East, in Israel, in Iraq, in places like that. And more recently, it has been demonstrated that they were also present further east in Central Asia, as far as the Altai mountain, where they expanded sometime. So for a long time, we had a rather, I would say, almost naive picture of this invasion and replacement. And what I want to show you today is what have been the recent advances in the last, say, 10 years, showing a very different picture from what we had imagined for a long time. So first of all, as I said, we start finding hominins outside of Africa looking like Homo sapiens about 180,000 years ago. This is one example of a specimen found in a site in Israel, in the Mount Carmel near Haifa. And it's a maxillar bone with a morphology that's clearly not Neanderthal. And so it shows that already in the past there has been several sort of intrusion in this gate to Eurasia, that is the Levant, coming from Africa. But the big expansion started much later, about 50,000 years ago. And 50,000 years ago, we are going to start to see Neanderthals, Homo sapiens, expanding in the mid-latitudes of Eurasia. This is how this process was seen not long time ago. You see, that's a publication of 2014, so we're talking about 10 years ago. And it's a simulation by a geneticist about how these things could have happened. The numbers that you see are numbers of generation, and you see that starting with an entrance in the Near East, there is a sort of wave expanding north first and then west, and replacing all the Neanderthals. So I would say that was the model of the big wave of modern humans replacing the Neanderthals. Unfortunately, we did not have much fossil hominins to document this process, and people were mostly using simulations like what I showed you, and also archaeological evidence. And in this time period, we have in Europe the spread of lithic assemblages, stone tools like these ones, something we call Upper Paleolithic, and so there are different names to these assemblages that is given, especially one thing we call the Aurignacian complex. And these stone tools are very different from what the Neanderthals were making in Europe before the arrival of Homo sapiens. There are stone tools that are very standardized. They are not very large in size. Many of them were probably part of missile weapons, like this spear that he showed on the upper right of this picture. And so archaeologists has been mostly supporting the idea that the spread of Homo sapiens in Eurasia was documented by the spread of the Aurignacian complex about 40,000 years ago or later, 38,000 years ago. And again, these kind of stone tools were in sharp contrast with what the Neanderthals have been making before, with very few standardized objects like this one, but a lot of flakes and stone tools that were, I would say, almost shapeless. Of course, prehistorians, they built up a whole typology of these objects, but I'm not sure the Neanderthal would have had the same typology of these objects or had the same ideas about how they had to be used. So that was the simple model of the Neanderthals making middle Paleolithic tools being replaced by sapiens, making upper Paleolithic tools like the Aurignacian objects, and the change occurring around 40 to 38,000 years ago in Western Europe. But actually, things were already a little bit more complicated, because there were sites where objects looking like the Aurignacian object were found, but they were associated with Neanderthals. And of course, that was a bit disturbing, because this did not really fit this simple picture. And these assemblages were found especially in Western Europe. This one that I show you here, this example, is an assemblage that we call Châteaubriand, that is found in center France mostly, in several sites. And it includes not just stone tools looking like the Aurignacian stone tools, but also bone tools and also body ornaments like this one. And as a matter of fact, a series of assemblages like the Châteaubriand has been found in different parts of Europe between the layers of middle Paleolithic stone tools made by Neanderthals and these Aurignacian stone tools made by the modern humans. And all these assemblages that has been found in different parts of Europe were all from a period between, say, 46 and 40,000 years ago. So they would predate the origination wave of Homo sapiens coming into Europe, and they were called transitional assemblages. So as you see on this map, there are many of them. There are in each region, they have been given names. They don't last for a long time. They last 4,000, 3,000 years. And the general wisdom was that these assemblages were made by the last Neanderthals before the arrival of Homo sapiens. It was not very clear why Neanderthal would start to make these things suddenly just before Homo sapiens would arrive. Some people would argue that they sort of invented their own Upper Paleolithic at the same time that modern humans were moving into Europe. But many people argued also that it was because at distance somehow there were a diffusion of innovation coming from the sapiens into the Neanderthal world that would trigger this evolution. And just to give you another example, this is an assemblage called Uludyan that is represented in Italy and Greece, mostly in southern Italy. And Uludyan has been considered for a long time like a sort of Italian Châtelperronien. You know, it's about the same story. We have stone tools that are sort of intermediate between the middle and the upper Paleolithic. We have bone tools. We have body ornaments. And it was believed to be made by the last Neanderthals. And in 2011, this picture started to be challenged. And this is a paper that has been published by an Italian colleague called Stefano Benazzi and his collaborators about discoveries in a cave in southern Italy called Grotta Cavaglio near Tarento. And what Benazzi showed in this paper is that actually there were teeth like that one that were associated to the Uludyan that were teeth of Homo sapiens and not of Neanderthals. So you start to see that the situation became much more complicated than what we had in mind before, this simple wave replacement. Another thing that happened more or less at the same time is that another of these assemblage found in Eastern Europe was connected by several archaeologists with discoveries occurring in the Near East, and especially with material from a site in the Negev desert, in a site called Boker Tartit. And this assemblage, that was also in this kind of ensemble of transitional assemblages, seemed to show how one could evolve from the kind of industries produced by Homo sapiens before leaving Africa and the Upper Paleolithic of Europe. And so people start to argue that maybe this also was something made by modern humans before the start of the origination, before I would say, the official wave of peopling. And this is what led us to start excavation in a site in Bulgaria called Bachokiro. Why we were interested in this site? Because there is this kind of assemblage that I just showed that seemed to be a sort of initial Upper Paleolithic preceding the origination. We have been very lucky because the site, although it has been already excavated by others before, was still in rather good condition. And we could excavate layers that were extremely rich in archaeological material and also in organic material. And we found this kind of initial Upper Paleolithic stone tools that has been found by others in the Negev desert or in Lebanon or in Syria. And of course, what we wanted to identify in Bachokiro is who were the makers of this assemblage. And we're lucky to find some hominin pieces. This is a human maxilla, and morphologically, this is without doubt a maxilla of modern human, a maxilla like your maxilla, and it's very different from what you would find in Neanderthals. So that was another, I would say, stone in the garden of the transitional assemblages, because once more it was shown that one of these transitional assemblages was not made by late Neanderthals, but by early modern humans. One thing we did in Bachokiro is using a technique called ZOOMS, zooarchaeology by mass spectrometry, to extend our fossil record of hominins. What is ZOOMS? It's a method to analyze fragments of bones like these ones, which are, I would say, shapeless. This is the kind of material that you find in abundance in archaeological sites. Actually, more than 70% of the bones going out of an archaeological site looks like that. In principle, there is nothing you can do with this. It's very difficult to identify what species belong to what species belong all these fragments. But if you extract pieces of collagen from these bones by taking a tiny piece of bone, dissolving it in an acid solution, and then cutting this piece of collagen with enzymes, and passing that in a mass spectrometer, you can weight these pieces of collagen, and using this technique, you can rather quickly and for, I would say, a low cost, you can identify what are the species to which all these pieces of bones belong to. And by doing that in Bachokiro, we could identify a series of supplementary human remains. And when we had these tiny fragments of humans, which of course are not very spectacular, there are a number of things we could do with them. First of all, we could date them. And these are direct dates that has been obtained on these human fragments. And you see that most of them, we are talking about human fragments, are about 45,000 years ago in terms of age. So much older than the origination wave. And what was most surprising is that these dates are much older than the dates of the last Neanderthals that we have further west in Europe, which are represented in blue. So it means that several millennia before the last Neanderthal disappeared in Western Europe, you already had modern Homo sapiens in Bulgaria making these kind of assemblages. And it was possible also to extract DNA from these people. And that was very interesting because, of course, it confirmed that we were dealing with modern humans, very ancient modern humans. And you see, that's a representation of chromosomes, a little bit like what I showed you in one of my first slides. You see little bars, blue bars, which are the equivalent of the red bars that I showed you in the previous slide. So this is integration of Neanderthal DNA. And this integration of Neanderthal DNA is something that occurred when these people, they left Africa and entered the Levant, the Near East. But you see that some of this inclusion of Neanderthal DNA are much bigger in size than these small bars. And they are much bigger in size because they correspond with an hybridization event that is much more recent in time than this early integration. So in other words, these people had a contact with Neanderthal several millennia before they arrived in Europe. But then in Europe, in Bulgaria, just a couple of generations before they lived, something like 5 to 7 generations, they had again an hybridization. they had also a hybridization event with Neanderthals. So virtually, we are dealing with the people who were the first in contact with Neanderthals in Europe. And what you see also in this distribution of Neanderthal DNA is that you already can identify the deserts of Neanderthal DNA that I mentioned previously. So the parts of the DNA where there is never any Neanderthal DNA in the genome of present-day humans, which means that the selection that sort of clean off our DNA from Neanderthal introgression in some parts of our chromosomes was extremely brutal and acted already in the first generation after the hybridization event. At the time when we published all this, there was another publication that was done by another group about a woman discovered in the Czech Republic, a skull of a woman, about the same age that Bachokiro, and also proving that this woman was a modern human. You see that she has some kind of African features. The interesting thing is that her DNA was quite different from the one of Bachokiro, suggesting that we had different groups of modern humans moving into Europe in this time. And also another difference in that in this case, there was this chunk of Neanderthal DNA documenting a recent hybridization. So it means different groups, some groups having contacts with local Neanderthals and some others having no contact with Neanderthals at all. And last, I want to show you another transitional assemblages that is called Lincolnian Ranisian Germanovician. I will call it LRG to simplify. So LRG is represented in Northern Europe, Northwestern Europe between British Islands, Germany and Poland. And this also, it's an assemblage that has been long attributed to the last Neanderthals. And we wanted to know if this was the case. So we resume excavations in one of the main sites of the LRG has been identified. Is the site of Ranis that gave the name of Ranisian. It's in Thuringia in Germany. It's really the heart of Germany. And the site is under a castle. You see the cave. Actually, it's a collapsed cave, so the excavation was a little bit complicated, as you'll see. Because it's a collapsed cave, we had to make a shaft through the collapsed roof of the cave. It's an 8-meter deep shaft to be able to excavate the archaeological layers that were underneath. The site had been excavated already in the 30s, but then no humans were found. And during our excavation, with a lot of efforts, using, again, this molecular technique that I mentioned, zooms, were able to identify a series of hominins in the site. We also explored the collections that has been left by the previous excavators of the 30s in the Museum of Halle in Germany. And altogether, we managed to obtain 12 hominins from this site. So that's another transitional assemblages for which we can document hominins. And again, doing ancient DNA, we were able to prove that these people were modern humans, Homo sapiens. And the date of these humans, it's even older than the dates of Bachokiro. So now we are dealing with hominins in a range of age beyond 45,000, between 45 and 47,000. And of course, all these discoveries change considerably the picture we have of this peopling of Europe by Homo sapiens, because the one-wave model, with the origination coming about 40 to 38,000 years ago was completely falsified. Basically, what we showed is that several of these transitional assemblages were actually not produced by the last Neanderthals, but by Homo sapiens, and that this event of intrusion of groups of Homo sapiens in Europe was something much older than everything that has been envisioned before. The other thing that's really interesting about the work in Bacciocchiro and in Ranis is that people more or less had this idea that the reason why Homo sapiens moved into Europe was connected to some kind of environmental or climatic change. Because this is already what triggered the Out of Africa movement. It's environmental changes that actually pushed out of Africa some population of Homo sapiens. So the general idea was, yes, these guys are of African origin, they lived in the Near East, so they must come into Europe by the east and southeast of Europe, and they probably come into Europe during a warmer episode. And actually people try to modelize that. What we showed in Bacciocchiro and in Ranis is that, in fact, they did not come at all during a warmer episode. What you see here is a tooth of a horse, and by sampling the enamel along the horse tooth, that is a tooth that is continuously growing during the life of the horse, so you can basically document chemical changes during a year and a half or something like that along this tooth, and measuring stable isotopes of oxygen, it's possible to reconstruct the mean temperature at the time when this horse lived in different periods. And you get a curve like this one showing the change of the mean temperature during the life of the horse between summer and winter. And that's really interesting because it gives you a direct thermometer to measure what was the temperature when people lived in the site, because it's the humans who killed the horse and brought the horse in the site. And doing that, what we showed is, for example, in Ranis, is that in Ranis, when these Homo sapiens coming from Africa reached Thuringia, the mean temperature at some point in winter was 12 degrees Celsius lower than the mean temperature in winter of Germany today. So I don't know if you spend night outside in Germany in winter, but you don't want to experience 12 degrees Celsius lower than present day. So these guys actually moving from the Near East, moving to Europe, during very harsh climatic conditions. They lived in the landscape that was the landscape of northern Scandinavia today, at the limit of tundra, the limit where you can still find some trees. And it's quite interesting because it shows something that was completely unexpected. And this thing is that Neanderthals survived a long time after the arrival of these people. We have a discrepancy in age between the last Neanderthals and the first Homo sapiens of maybe 6,000 years, which is a very long time. But Neanderthals basically lived where the conditions were the best. They lived in southwest Europe. They lived in southwestern France, for example, where there are many sites of this time period. And Homo sapiens start to colonize by small groups the marginal areas, the areas in the east and in the north that were not very favorable for humans. And they were probably more resilient than the Neanderthals and more able to deal with these local conditions. And the other thing that we discovered is that all these groups of humans from Bacciocchiro, from Zlatikun, from Ranis, were different groups of humans, were not the same group. And there were groups, that's what geneticists say, of very small size, a couple of hundred people. So rather than a wave, what we have is sort of small wavelets of humans coming and settling at the periphery of the Neanderthals in the less favorable areas. And it's only much later than the real origination wave is going to come, is going to replace the Neanderthals, and also, by the way, is going also to replace these pioneer groups. So the evolution of our knowledge on this question in the last, say, 10 years is that, first of all, modern Homo sapiens were already present in Europe much before what we imagine, at least 7,000 years before what was believed just 10 years ago. So we're talking about 47 years ago. So many of these assemblages like Bachokirian, like LRG, like the Uluzian, like Boinician is another one, that were believed to be made by last Neanderthals actually were made by this pioneer population of Homo sapiens. Of course, it raised the question of, did any transitional industry ever exist? I mean, because this notion of transition was that the Neanderthals were sort of experiencing their own transition to the upper Paleolithic, but finally it looks like no. They just continue to do their middle Paleolithic before being replaced by these groups. And of course, there is this very puzzling statement, observation, that all this lasted a very long time, that there is several millennia before the first intrusion of sapiens small groups and the extinction of the last Neanderthals. There are still pending issues, and I don't want to hide them. One issue is that there is one site in France where people have argued that maybe there is even older sapiens, maybe before 50,000 years ago. And we have still the issue of the Châteaubriand that looks like the origination and has been made by Neanderthals, has been associated to Neanderthals. And so that's a kind of stone in our shoe, because it does not fit all this picture that I gave you today. But recently, just last year, we published a small piece of pelvis that has been found in a Châteaubriand layer. And this pelvis, a pelvis of a baby, has a morphology of sapiens. And so it's possible that the Châteaubriand actually is some kind of assemblage made by a mixed group incorporating some Neanderthals and sapiens. At least we know that there are Neanderthals around, because there are a number of Neanderthal remains found in these sites. But now we have also sapiens, so the picture might become even more complicated in the future. And the second issue is what kind of interaction sapiens and Neanderthals had. I mentioned already this episode of hybridization, but there are some troubling observations, like, for example, the occurrence of body ornaments that are very similar in the sites where the last Neanderthals has been found, and the kind of body ornaments that are being produced by the first Homo sapiens. So it's very likely that there are not only gene exchanges, but also innovation passing from one group to another. So you see that the reality is, in general, more complicated than what we imagine as archaeologists or paleoanthropologists. And of course, all this work has not been made by myself alone. And here are the list of people who contribute to the work in Bachokiro and in the Ranis. Thank you for your attention. If we have some minutes, I will be happy to address any question by the audience.
Prego, c'è una domanda lì.
Thank you, that was wonderful. I learned so much. And I was really intrigued because you were saying that 2% of Homo sapien DNA is Neanderthal DNA. And you could do that because there are populations in Africa that didn't have contact with Neanderthals. Is it possible to speculate on the opposite? Is it the case that 5% of Neanderthal DNA is Homo sapien DNA by the last Neanderthal remains that are found?
That's
because you tend to think that hybridization is asymmetrical in terms of the potential survivability of offspring.
I tell you what the issue is. Homo sapiens post-contact with Neanderthals, there are many, 8 billion. So you can explore their DNA and try to find trace of Neanderthal DNA. Neanderthals, post-contact, we don't have any at the moment. I mean, maybe we have some, but they have not been investigated for ancient DNA. So my prediction is that the day we'll do it, we'll find that. And I want to add something else. I mentioned that the flow of sapiens out of Africa started 180,000 years ago. Actually, there are genetic evidence that there was already more ancient pulses. And in particular, paleogeneticists have argued that among recent Neanderthals, the Y chromosome and the mitochondrial DNA is one part of the genome that's transmitted just by the females to their descendants. So the Y chromosome and the mitochondrial DNA of Neanderthals in Europe has been replaced by Y chromosomes and mitochondrial DNA of African origin. So that would be an example of reverse, I would say, integration of African DNA into the Neanderthal genome. And that's something that's quite old because we don't have a precise age, but it could be 200,000, 300,000 years ago.
C'era un'altra domanda. Ah, stimolato, vedo. Prego. Dopo lei.
Can you hear me?
Yes.
I was wondering, what are your definition for saying is one sort or the other? We are all human, you just mentioned. So what criteria you use for say, we are human or we are not human, first of all. And secondly, I was wondering why you want to make this big difference and what are really the reasons for, if you mentioned it's not environment, for this movement, for this going away, and then the hybridization was probably, you said, small groups that chase the other one, etc. And why did it finally make cities out of all these groups?
Oh, there are several that deserve a whole lecture. No, but okay. Definition of humans, it's a complicated story. As I said, humans, today they know what are the humans. The question becomes more difficult when you move back in time. And frankly, I think there is no border between humans and non-humans. So, okay, we can have this discussion later.
Well, yes.
But what we can do, and it's quite simple, is to identify Neanderthals from Homo sapiens, genetically and morphologically. So it's not like comparing, people living, I don't know, in Spain and people living in Iraq, people living today, they are all the same. Neanderthals and sapiens, they are quite different. They are different anatomically and genetically. We can identify them quite easily. Now, why there was this replacement and why it's not climate? What I think is that all these hominins, they were evolving toward more complex behaviors in terms of technology and also in terms of social complexity. But at the end, our species went a bit faster. And going a little bit faster created a difference. Maybe it was not a huge difference. As you saw, it took millennia before the replacement took place. But there was enough difference for one group to replace not just the Neanderthals, but actually all the others, all the other hominins that exist in Eurasia, in the islands, etc., they were eventually replaced by sapiens. So there is something with sapiens that makes it different. And the fact that we find them in very difficult environment show that again, is not the climate that pushed them there, is their ability to cope with this kind of environment. And their ability to cope with this kind of environment, I think, is both technological and also probably social. So humans are very good at creating networks of individuals that can cooperate, and not just an extended family or a small group of individuals, but you can cooperate and you can find some kind of solidarity with people that you will never know, that live very far, but with whom you share all sort of features and things like that. And last, about the hybridization, well, it depends. If you like Dino Buzzati and you read Un Amore, you can think that they had some kind of romance between Neanderthals and sapiens. I have another story. It's a story of a group of sapiens coming close to the cave of Bachukiro, where there are groups of Neanderthals living there, and they kill them all. But they keep 2 women. And if you wait 5 generations, they will have the kind of genome that I showed you. So you can choose. I think there is no arguments to choose one or the other. I think probably both story occurred in the past. Voilà.
Abbiamo ancora solo 2, anche qui volevate? No, ancora questa è l'ultima, grazie.
Thanks for the great talk. I would like to know, is there a possibility to know the size of these populations? How many people did it involve?
Yeah, well, demography for the past is notoriously difficult. There are 2 ways to go. One way is archaeology. And so archaeology, how you do that, you have to make some assumptions. And first of all, the assumption is that the group of hunter-gatherers cannot be less than 20, 30 people, because if they are much less, they don't survive. But they cannot be hundreds of people living together with the type of economy that they have. So already you have one parameter. And then you can look at the territory of one group. And this is kind of easy, because you look at the raw material that they use and how this is dispersed in the landscape. And so you know that they need a territory that is something like 80 kilometers in diameter, maybe 100 something. And so then it's easy. You take a map of Europe for one of these assemblage, and you see how many circles of 120 kilometers you can put one beside the other. And then in each of these circles, you put something between 50 and 150 people. And what you find is that some of these assemblages, they must have been something like between a couple of 100 people and maybe 3,000. This is the kind of size we have. Then you can go for genetics. And genetics gives you an idea of the variability, the diversity, the genetic diversity in the group. Well, it's complicated because not everybody has a descendant, and there is something that the geneticist measured that is called the size of the effective population. That means the number of individuals that you need, breeding individual, to explain the variability, subsequent variability. It's not the real size of the population, but it gives you an idea. And putting all this together, yeah, the idea is that Neanderthals in Europe, there were a couple of thousand people, something probably less than 10,000 for Western Europe. Think on that, is 2 or 3 big villages of today dispersed on a huge territory. And these groups, like, I'm a bit embarrassed to speak about Rennis because there is another paper coming that is not published yet about the genetics, but I can sort of spoil a little bit the conclusion. Yeah, the estimate is that there are maybe something like 300 people. the Ranis, Zlatikun, because there are genetical connection between Zlatikun and Ranis, that, by the way, it's amazing that we can connect different sites in this time period, and we're talking about very small groups. So to me, it blows my mind, because it means there are groups of a couple of 100 people leaving a place like Syria, making their way throughout Western Eurasia as far as Thuringia, you know, and settling there for several centuries or millennia. And then you have another group independently moving from the same kind of region, going to Bulgaria, going to different places like that. And so it's a gradual sort of filling of the landscape with these small groups. And in the meantime, you have the Neanderthals having the last Neanderthals having their happy life in Périgord. They don't know what's going to happen to them, but they are going to disappear a couple of millennia later, when the real wave is going to come, which is the wave of the Aurignacian. And by the way, we have today in the genome of present-day Europeans, we still have a bit of the DNA of these sapiens moving into Europe with the Aurignacian, but not from these guys, from Ranis and Bachokiro and etc., which means that the first arrival did not really make it. You know, they arrived, they lived there, they settled, but there is no real continuity with the subsequent population. And that's really interesting, because it's something that you can maybe imagine for other areas. Like, you know, there are a lot of discussion about how Australia was peopled, how America was peopled, and this notion that the pioneer population, they make it, they leave some archaeology, and so archaeologists say, wow, we have the oldest Australian, but maybe this Australian, they have no descendants today. And the real peopling of Australia is something which is unrelated to these very early sites.
Thank you. You've been so interesting that we have one last comment. I couldn't say no.
Birgit Eriksen from the General Prize Committee. Thank you, first of all, for an absolutely brilliant presentation. What you have made clear as the other prize receiver from that generation, Eske Willerslev, who is also sitting there, that to build bridges between humanistic and natural sciences can create absolutely fantastic and fascinating results. Yeah. And I'm sure the audience here grasps that and understand there is a great world of new ideas and discoveries that can be made in the cross area between natural and humanistic sciences. Thank you very much.
Thank you. And this is a very important remark, because, and I would like to end with that, because if you leave me the microphone, I'm going to keep people on. But there's really something important to, if you want to just remember one thing leaving this room, is that dealing with human evolution, you have to consider cultural evolution and biological evolution together, because there is a permanent interaction between the two. So our biology is shaped by behavior, economy, the way we mate, all these kind of things. It shaped our biology. And in return, this biological change allowed a lot of changes in the behavioral sphere. And so it's a real mistake to disconnect the two, and you always have to consider the two aspects of the human history.
That's right. Thank you so much.
Thank you.
Bene, io vi ringrazio e all'anno prossimo. Grazie.